Okay, thank you very much. It's very nice to be here. So what I'm going to be talking about today is why it is within insect populations you get genetic variation in susceptibility to infection, and particularly susceptibility to virus infections. So here's just an outline of what I'll be talking about after some extremely brief background. I'll then look at several different aspects of this genetic variation. So firstly, I'll look at whether or not the genetic variation has a simple or a complicated genetic basis. So in other words, are there many genes contributing to the genetic variation, or are there just a few? Secondly, I'll look at what evolutionary processes are maintaining this variation. Obviously, the susceptible individuals are more likely to get sick, they're less likely to survive and reproduce, so you'd expect the susceptibility alleles to be eliminated by natural selection. So why do you get this maintenance of so much genetic variation and susceptibility? I'll then look briefly a little bit at the genes involved in this variation before talking about the role that bacterial symbionts have in controlling susceptibility. So just to start with a bit of background. The background, all I want to say really is that you can go to populations of pretty much anything. It could be plants or insects or vertebrates, and you find lots and lots of genetic variation in susceptibility. So here's just a data set from humans that I'm quite fond of. It's a compilation of twin studies. And what people did was they took pairs of twins where one of the twins had become infected with an infectious disease and asked what's the probability that they infect their twin. And this just compares the rates of transmission between monozygotic twins, which are genetically identical, and dizygotic twins, which are obviously just siblings. And you can see transmission rates are really spectacularly higher amongst genetically identical individuals than amongst normal siblings. And that suggests there's really a very substantial genetic component in humans in susceptibility to these 4 different infectious diseases. So I could have shown you data from pretty much anything. Here's another example with lots of genetic variation in susceptibility. These are some Drosophila lines. This is a bar chart. There's about 200 highly inbred Drosophila lines. And we've assayed each of these for the ability to infect them with a virus, which I'll talk about later. And this is a proportion of flies which became infected in our experiments. And this was a big experiment. There's about 10,000 flies there, so the lines are very well, the estimates are very good. And you can see that some fly lines are almost totally resistant. Basically, we can't ever infect them with the virus, and others are extremely susceptible. So there's a full spectrum there from extreme resistance to extreme susceptibility. And that's typical of most of the systems which we look at. In insects, this might be important in many different contexts. Obviously, there's beneficial insects like honeybees, which are suffering from infectious diseases, often viruses, which might be impacting their populations. People might be interested in, for example, selective breeding to produce more resistant honeybees. We work on disease vectors like mosquitoes, and in biological vectors where the pathogen replicates within the insect, you find a great deal of genetic variation in the immune system of these insects, which will control the ability of the mosquito to vector disease amongst people or vector diseases amongst other species. And finally, of course, if you're using pathogens in biological control, then here this genetic variation might determine the success or failure of your control program. So we're interested really in a couple of questions about this variation. The first one is one of evolutionary process, which is why is all this variation there in the first place? Why isn't everything simply resistant? And secondly is a slightly more mechanistic question of what it is that's causing this variation at the genetic and molecular level. So to take the first of these questions, does this genetic variation have a simple or a complex genetic basis? So by that I mean, are there lots of genes contributing to it or just a few? And several things matter here. The first thing is when you look at a particular gene and you look at the 2 alleles of that gene, what are their effect size? So is that gene having a small effect on susceptibility or a large effect? However, you might have a very major effect polymorphism, but if it's at a very low frequency within the population, it's not going to contribute much to genetic variation. So the second thing which matters is the frequency of alleles within populations. So if you have major effect things which are common, then you're going to have a very simple genetics. And finally, of course, a number of genes with these variants. And if you look across quantitative traits in general, you tend to find that the rule is that they tend to have a very complex genetic basis. So when people, the best studied ones, tend to be non-communicable disease in humans, and here you find very large numbers of genetic variants contributing to the genetic variation. And each one of those genetic variants typically has an extremely small effect on your chances of getting the disease. So I'm interested in whether or not susceptibility to infection might be different from this rule for quantitative traits. And I'd argue that it might be, because you have different types of selection pressure acting on susceptibility to infection than you do for other quantitative traits. So underlying our work on this variation is the ability, firstly, just simply to find these genes and sort of stamp collect lots of resistance genes. And we use a variety of different approaches. I probably started my interest in this, 6 or 7 years ago. And back then, we mostly started by doing linkage mapping. So here we just took an inbred line or isogenic fly line which was resistant. We'd find one which was susceptible. We'd cross them. We'd generate recombinants and make those themselves isogenic or homozygous. And then we'd score molecular markers across the genome, measure resistance, and look for which bits of the genome contain the genes controlling resistance. And then there's lots and lots of tools in Drosophila which allow you to map these genes with more precision. However, even in Drosophila, this is an extremely difficult and slow and tedious process, even when you've got all those genetic tools available to you. So this works perfectly well, but it's very slow, and it generally only works for fairly major effect genes. The second approach we've used is using panels of lines which have been generated from advanced intercross populations. So here there's a set of 8 inbred founder lines chosen from across the wild, so they contain lots of the genetic variation that's present in Drosophila melanogaster. All were just sort of bunged together in a population, left to go for about 50 generations. So then you have a population where all the chromosomes are a fine-scale mosaic of those 8 founders. And then from those recombinants, they were inbred to generate a panel of 800, or in total 1,500 recombinant inbred lines, each of which has a fine-scale mosaic of these 8 founders. So the 8 founders have complete genome sequences, and then you can score SNPs at a high density across these recombinants, and you can effectively statistically reconstruct the genome sequence of all of those recombinants. So from this you can then map with great precision quantitative trait loci to very small regions, and because you then have 8 founders and their genome sequences, you can divide those 8 founders into the resistant and susceptible groups, and from the genome sequences you can get fairly small lists of the potential genetic variants causing that resistance. The final approach I've taken is using whole genome association studies. Partly we've used panels of inbred lines with complete genome sequences. We also do a lot of genome sequencing ourselves when we're looking at species other than Drosophila melanogaster. Here you just have all these genome sequences, you measure resistance, and then you look for SNPs across the entire genome which are associated with resistance. If we have species other than Drosophila melanogaster, we just infect the population, we take the extremes of susceptibility and resistance, and then sequence those pools of extreme individuals, and again look for differences in the frequency of SNPs. So here the advantage is that you basically often end up with the exact causative variant, or you end up with a very, very small list of genetic variants which could be causing it. So it looks in some ways like, as you move across here, things get better and better because you get better and better resolution down to the gene or the nucleotide level. But there's a cost as you move over here, and there's about 5 or 6 million SNPs that we're using here. So you've got a big multiple testing problem. So you need extremely small p-values, and you need extremely large experiments to get those. And you also need fairly large effect genetic variants. Whereas over here, this problem is much less. So there's a sort of trade-off between the resolution of your study and its statistical power. Okay, so I won't really talk much more about the methods we've used. So we then needed some traits to measure, so we used viruses. I'll talk here about these 5 viruses here. They're all RNA viruses. These 2 here are negative sense RNA viruses. These are all positive sense RNA viruses. They're taxonomically fairly diverse: rhabdovirus, dicystroviruses, nodoviruses, different families. But Perhaps the most important thing for this talk is that 2 of the viruses were isolated from Drosophila melanogaster, so they have a co-evolutionary history with Drosophila melanogaster, and the other 3 were isolated from different species, so there's been no co-evolution between these viruses and Drosophila. So we took our panel of inbred fly lines, we infect them with the viruses, and we can calculate the heritability of resistance, or the amount of genetic variation that there is in populations. And that's shown here. You've got 5 different viruses, and you can see there's quite variable amounts of genetic variation there. For 2 viruses, there's lots of genetic variation, and for 3, there's much less. And these 2 viruses are the viruses which naturally infect Drosophila melanogaster. So that sort of suggests that maybe this process of coevolution between Drosophila and the virus is increasing the amount of genetic variation that's present in populations. So these fly lines which we'd infected have complete genome sequences, so we can just look across the genome, do an association study. So this is a position across the genome, across the 5 chromosome arms, and this is the p-value. And you can see that find an extremely small number of extremely strong associations. So for example, the p-value here is 10 to the minus 20. So there's just one variant there which has this extremely large effect. And the same down here for this sigma virus. So there's very strong associations there, and there's an extremely small number of them. But that's only the case when we look at the viruses which naturally infect Drosophila melanogaster. If we take viruses from another species, even if they're quite closely related to the virus which we got from Drosophila melanogaster, we basically find none of these associations at all. So you find major effect variants, but you only find those major effect variants when you're looking at viruses which naturally infect Drosophila. So one of the problems with association studies is you tend to get these associations, and it can be very difficult to verify them, and that's often because people are working in things like things like humans. So the nice thing about Drosophila is we can experimentally verify our associations. We did this a few years ago, and the approach we took was I'll explain here. So here's an example where we've just zoomed in on one of the plots from the previous slide. You've got the position across the genome. Each dot is a SNP. And you've got this cluster of strong associations with resistance to the sigma virus here. And in the middle of them, we've got a polymorphic transposable element insertion. So our obvious explanation is that the transposable element insertion is what's causing resistance, and then you get a few other SNPs nearby, which are in linkage disequilibrium with that transposable element. So what we did was we took a back clone of this region, which happened to have the transposable element insertion. So this is a large clone of about 40,000 base pairs of that region of the Drosophila genome. Then in E. coli, we can precisely excise that transposable element, leaving otherwise identical backs, but just missing the transposable element. We can then pop those into the same position in the genome of Drosophila in a mutant which lacks a gene which is affected by this transposable element insertion. Then we infect them with the virus, and rather pleasingly, we recapitulate the variation which we saw in nature. When you've got the transposable element, you have much lower viral titers than when you don't. And we've now repeated this for all the genes which we've found, and we've basically been able to verify all the results of our association studies. So rather unusually for a Drosophila lab, we do occasionally venture into the wild, and we're sort of somewhat aware that Drosophila does exist in people's dustbins as well as in our lab. So we went to central Athens on a 3rd floor balcony of a flat, and we collected wild Drosophila. And in this case, we measured this, the transmission rate in wild caught flies of this virus called the sigma virus. And here we've got a polymorphism in the gene called P62, which is controlling susceptibility in our association studies. And what you can see is that in nature, we also see an effect of this polymorphism on the transmission rate of the virus. So these associations matter not only in our lab, but they also matter for natural populations. So going back to the original question of how complex the genetic basis of susceptibility is, we can quantify this by taking our polymorphisms and looking at what proportion of the total genetic variation that we see is explained by individual genetic variants. So if we look at the virus called Drosophila C virus, this was affected by this gene here called pastoral. And in this population, pastoral explains about 50% of all the genetic variation that we see, We've recently done a sort of comparable experiment with a different population, and here we found that this gene explained 85% of the genetic variation. Now, if you've ever sat through a talk by someone doing association studies on humans, on non-communicable diseases, they'll often put up variants which explain 0.4% of the genetic variation. So these really are whoppers, they're really big effect. These other three genetic genes affect the Drosophila sigma virus. Here we've got a gene called Chekhov1 that explains about a third of the genetic variation. This gene P62 is about 8%, and this other gene has rather little effect. But the effect of all these alleles is extremely large, so they're all making flies extremely resistant. And the difference in the proportion of the variation explained is basically attributable to their frequency within populations. So these two genes here are fairly low frequencies, and this one here is a bit more common. So we seem to be seeing a really major, really simple genetics here, with much of the genetic variation explained by a few major effect genes. And then in the background you have a sort of polygenic genetic variation as well. So what about other insects and parasites? Well, the other species which we work on quite a lot is this mosquito called Aedes aegypti, which is a disease vector in humans. And we've been working on filarial nematodes, and particularly this filarial nematode Brugia malayi. So although Aedes is an important disease vector, it isn't actually the major vector of filarial nematodes, but it's a useful sort of lab model. So Brugia malayi is a cause of lymphatic filariasis, which is this disease in humans. So we've got two lines of Aedes here, and this is just a count of the number of worms which develop, and you can see there's lots of worms develop in this line here, and rather few worms develop here. So two lines which differ a lot in susceptibility. And if we do a cross, we can map this cross back to the sex chromosomes. So basically, this is repeating a result which was previously done way back in the 1960s. And this is a cross where the only difference between these two genotypes here is which parent their sex chromosomes came from. And you can see that basically all the difference in susceptibility is caused by the sex chromosomes. So we wanted to do some association studies here. And the problem that we have is we now have a 1.4 gigabase genome, which is about 10 times the size of Drosophila. And it becomes sort of prohibitively expensive for us to sequence the entire genome. However, much of the genome in a big genome like that is repetitive stuff that doesn't really do very much. So instead we decided to focus on the important bits, or some of the more functionally important bits, which is a bit of the genome which encodes proteins. So instead we just resequence the exomes of these mosquitoes. And you do that just by synthesizing probes which map the entire exome, and then you capture that bit of the genome. So now we're sequencing a 10 megabase genome rather than a 1,400 10 megabase exome rather than a 1.4 gigabase genome. And then all we did was we went to Kenya, collected a bunch of mosquitoes, infected them with the worms, and dissected out individual mosquitoes to see whether or not the worm had developed, and then sequenced the exomes of those individual mosquitoes. When we did this, we get much the same result as we tend to find with Drosophila and viruses. We get this one extremely strong association here, again suggesting that pretty much all this variation is not only explicable by the sex chromosomes, but also by a single genetic variant. So that's my first conclusion, that in the systems we've looked at least, and I'd argue quite a lot of others as well, susceptibility to infection often has an extremely simple genetic basis, and that's quite different to what we see in many other quantitative traits. So the second question I want to talk about is what's maintaining these major effect genetic variants in populations. So obviously these viruses are pathogens, they make the fly sick when they get infected. So if you're susceptible, you're more likely to become infected, you're less likely to reproduce and survive. So you'd expect natural selection is going to eliminate the susceptible alleles, and all else being equal, everyone would be resistant. But of course there's lots of population genetic processes which can maintain genetic variation in populations despite that type of selection. The sort of ubiquitous and in some ways slightly dull explanation is it's simply recurrent mutation. we're all faced with a rain of mutations. You might get this rain of sort of deleterious mutations, making you more susceptible. And it takes time for purifying selection to remove those mutations. So this balance between selection and mutation will mean that most traits vary genetically. Also, genetic variation can be maintained by natural selection. For example, resistance to infection is quite often costly, so there might be some balance between the costs of resistance and the benefits of not being infected, and that balance of the costs and benefits can result in negatively frequency dependence, maintaining the polymorphism. Or finally, maybe we're looking at sort of transient mutations. So maybe mutation throws up new resistance genes continually through time, and natural selection picks up on them, drives them through to fixation. But this process is happening continually. When you take a snapshot in time, what you're seeing are polymorphisms as they're spreading through populations on their way through to fixation. So we're looking at transient polymorphisms. So which one of these seems to be true? Well, I've already given you a clue that natural selection might be important, in that when we look at naturally occurring viruses, we see more genetic variation, and we also see these major effect genetic variants. And we've sort of repeated this on more viruses than I've shown here. The pattern seems to hold up. So that suggests that when you have selection for pathogens, that's somehow inflating genetic variation, not removing it. Secondly, because we know the specific mutations which are causing the resistance, we can look at a closely related species of Drosophila, see what sequence it has at that position in the genome, and infer what was the ancestral state. And every time we find a resistance gene, we always find that the ancestral state is to be susceptible. Again, this fits with a sort of pattern where hosts are continually evolving new ways of becoming resistant in some arms race with the pathogens. It doesn't fit with a sort of selection mutation balance argument. Finally, we can look at linked genetic variation to the mutation or the polymorphism causing resistance and infer how natural selection has acted on those variants. So what these are here, this is two different genes. This is a gene called P62, which affects the Sigma virus. This is a gene called Chekhov-1, which also affects the Sigma virus. What it is, it's an alignment of polymorphic sites. So we've basically sequenced the gene. We've stripped out all the sites which don't vary, and these are just the sites which contain SNPs. At the top, you've got resistant alleles of the gene. At the bottom, you've got susceptible alleles. And what you can immediately see is there's lots of genetic variation amongst those susceptible alleles of the gene, but the resistant alleles of the gene are all extremely similar. And that tells you that they all shared a common ancestor very recently, and there's been insufficient time for mutation to cause genetic variation on these copies of the gene. And that's a classic signature of a partial selective sweep. So the resistance mutation has arisen recently, it's rapidly spread through the population, and there's simply been insufficient time for genetic variation to occur. So what we're seeing appears to be consistent, at least, with transient polymorphisms. And it turns out that other people's data have shown rather similar processes in the virus population. So this is data from someone called Annie Fleurier. And what she did was, from the early 1980s to the early 1990s, she went out into two populations in Germany and France and collected isolates of the Drosophila sigma virus. And she took them back to the lab and injected them into two stocks of Drosophila, which we now know differ in this gene P62, and asked whether or not they could infect the Drosophila. And what she found was that during this period, there was a sweep of a viral genotype which overcame the host resistance. And this happened both in the population in France here, and this population in Germany here. So our population genetics data shows these sweeps happening in the resistance genes, and this more direct observation shows a matching process happening in the virus population. So we seem to have a sort of process like this, where we have this arms race. You have the resistance gene sweeping through the fly population, and then this is followed by some sort of countermeasure arising in the virus population which overcomes a resistance, and this is sweeping through the virus population. So this directional selection on these major effect variants can also explain why we get such a simple genetic basis to susceptibility to viral infection. So what I'm showing here is a cartoon where I've plotted the frequency of alleles within populations against their effect size. And the typical pattern which people see in human association studies on non-communicable disease is this. So you have minor effect variants which are quite common within populations, and you have major effect variants, and they tend to always be quite rare. And this sort of, is it's not great for association studies because it means whatever you find, it's not very interesting. Because it's either so rare it's not very interesting, or it's either so rare it's not very interesting, or it's common but it's having such a small effect. It's also not very interesting. But the reason for this is that this genetic variation is occurring due to mutations which are deleterious. So the probable explanation of this is you get mutations occurring which make you more susceptible to heart disease or whatever. Effectively, they're just breaking some well-operating physiological system. If they have a major effect, natural selection is very good at getting rid of them, so you only ever find these at low frequencies. But if they have an extremely small effect, natural selection isn't very effective, and they become much more common. However, what we're seeing is directional selection pushing these new mutations increasing resistance up in frequency. So we find major effect variants which are very common. And that makes it, I think, it certainly makes it a much easier thing to study than these other traits, and possibly more interesting. So that's my second conclusion, that directional selection to increase resistance to infection can explain the simple genetic basis that we see to these traits. So next I want to talk just very briefly about the genes involved. We do some functional work, and I won't really talk very much about that because we're probably not very good at it. But I'll talk about a few generalizations which we might see when we study these genes. So the first thing we've looked at is when we find a resistance gene, is there just one genetic variant in that gene which is affecting susceptibility? Or are there recurrent changes to the same gene which are sweeping through populations, leading to successive increases in resistance? So it turns out that at least two of the genes we've studied, and the only two we've looked at in great detail, we actually find there's multiple genetic variants within the population. So this is most dramatically seen in this gene Chekhov-1, which makes flies resistant to sigma viruses. So the ancestral state was this. You've got three genes, and in the middle, this is Chekhov-1 and paralog Chekhov-2. They're just of normal intact genes. And then a transposable element insertion comes along and inserts in the middle of Chekhov-1, and that makes flies very resistant to infection. And that allele is now the commonest one within natural populations. But we also find a third rare allele where this new bit of sequence here has been duplicated twice, and that makes flies almost totally resistant to infection. So we see this sort of allelic series where each successive change makes flies more and more resistant to infection. We also see slightly less dramatic effects in another gene called Pastoral. This one affects Drosophila C virus. So here's our association study here, moving across the genome. There's a cluster of SNPs here which are associated with resistance. And we were interested in which of these SNPs cause differences in susceptibility. So we used this other panel where we had a set of 8 different alleles from different founders. We could assign them to different levels of resistance. And when we do this, we find 3 different alleles with differing levels of resistance. So we have a major difference here with 5 of the lines being extremely resistant, 2 lines being much more susceptible. But also these 2 susceptible lines also themselves differ in their susceptibility. So there seem to be 3 alleles here. When we sort of intersect these 2 changes, we find that the major change is caused by an amino acid substitution, which makes flies extremely resistant to infection. And then there's a sort of non-coding, possibly regulatory change, which has a smaller sort of additional effect to this of further increasing resistance. So it seems to be quite common that you have the same gene where you're having successive changes, each one increasing resistance. So what about potential mechanisms? Well, I suppose at the broadest possible level, you could imagine 2 ways to become resistant to a virus. The first thing is that you have lots of antiviral molecules and antiviral sort of processes in cells, sort of immune systems and restriction factors, which function primarily to protect you against infection by viruses. So it could be that you're taking your antiviral defenses and in some way making them better to protect yourself against infection. But also, the virus itself is going to be hijacking lots of your cellular machinery in order to promote its own replication and transmission. So you could also become resistant, rather than improving your antiviral defences, by instead altering this cellular machinery which is being hijacked by the virus in order to interfere with the viral replication cycle. And they produce slightly different sort of predictions. So the first one is that the susceptible allele here, if it has any effect, will tend to be antiviral, because you're taking antiviral defense and you're making it better. Whereas here, the susceptible allele will actually benefit the virus, so you're effectively taking this benefit away. So we can do this, we can look at mutants or knock down these genes, and what we find is that if we knock down the expression of the susceptible allele, you tend to become even more susceptible. So even the susceptible allele of these genes is making flies resistant, and the resistant allele just does this better. And we can also do the sort of reciprocal experiment. We can overexpress the susceptible allele. You overexpress the susceptible allele, again this makes you resistant. So it seems that you're sort of changing your antiviral defences somehow. So the third interesting thing is a slightly different situation is in this gene Chekhov-1, where we had this transposable element insertion I've mentioned a couple of times before. And here what we think has happened is that a gene which seemed to have no involvement at all in, or no effect at all on antiviral defences seems to have sort of given rise to a new gene and a new gene product which seems to have an antiviral effect. So the original gene here has 4 exons and produces this transcript here. You then got this transposable element insertion in orange, and here you get 2 different transcripts. So you get a short transcript here, and then you get a second set of transcripts which start in the transposable element and take in the rest of the protein. So you're effectively getting largely 2 very truncated versions of this protein, or of these transcripts at least, being produced. So what we did was we knocked out these different transcripts being produced, and we looked at their effect on viral susceptibility. And the interesting thing we found is, remember this product here makes you resistant, that if you knock out these transcripts here, you become susceptible again. So knocking out this gene has no effect, knocking out this transcript here has no effect, but knocking out this one makes you susceptible. So it seems that the transposable element insertion has given rise to a new transcript, and this new transcript has gained a novel function. And it really has, we have no idea what it's doing. So that was looking at a sort of phenotypic effect of viral infection. This is looking at viral titers. They both show the same thing. So a third, or I don't know, this is the fourth general property of these genes, is they tend to be rather specific in their effects. So if you take the resistant allele and you expose it to a different virus, it tends to have no effect on susceptibility. So at a rather broad level here, we have 4 different viruses, have 3 genes, the resistant allele in red, the susceptible allele in blue. These 2 genes make you resistant to this virus. This gene makes you resistant to this virus, but there's no cross-resistance at all. So at that broad level you see specificity. But also if you go into populations and you take multiple virus isolates, you often find resistance genes only affect certain viral isolates. So you've got 5 viral isolates here. This is a bar plot of 92 fly lines. We've inflected the fly lines with the virus, that's in this case the sigma virus. This is a gene called P62. And you've got a polymorphism here where you've got a single amino acid change which makes this fly line or makes the fly lines resistant to infection. But you can see that this polymorphism only affects this virus isolate here. It doesn't affect any of the other 4 virus isolates. So you get this sort of very precise specificity, and these are very, very similar virus isolates. They all shared a common ancestor, we think, probably within the last 100 years. So I won't really talk about the genes at all and what they do at a molecular level. I'll just give you the sort of outline. In DCV, this is a gene called Pastrel. It's on the surface of lipid droplets. Gene called G1 is in P-bodies, which are sites of RNA degradation, also the location of RNAi, which is the risk complex, which is the main antiviral defense of Drosophila. P62 is involved in autophagy, targets cargoes for destruction by autophagy. This is an important defense against rhabdoviruses. This is a rhabdovirus. And Chekhov-1, we have no idea what it does whatsoever. So that's my third conclusion. These genes have, the genes being changed, they're often existing some antiviral defenses. The effect of these polymorphisms is extremely specific, often down to particular genotypes of the virus. And when we look at the genes, we often find multiple alleles which have differing levels of susceptibility. So up to now, I've just talked about genes in the Drosophila genome that affect susceptibility. And finally, I just want to touch on the role that bacterial symbionts might have in affecting susceptibility to infection. So if you go to insect populations, you find a sort of bewildering array of bacterial symbionts which typically live within the cells of the insect, and their primary mode of transmission or sole mode of transmission is vertically, going maternally through the egg and the cytoplasm of the egg. So you get these heritable symbionts, and as a geneticist, they're sort of it's okay to study them because they're heritable. It means that from an evolutionary perspective, they can encode heritable traits, but outside of the Drosophila genome. So we became interested in these because it was discovered that some of them can protect insects against viral infection, and particularly this bacterium called Wolbachia. So Wolbachia is an extremely common symbiont of insects. It's typically estimated that maybe 40% of insect species are infected with Wolbachia at some level, prevalence. It's usually only maternally transmitted within populations, and it can provide resistance against viruses. And the reason that this became very topical is people transferred this bacterium into Aedes aegypti populations and found that it blocked the transmission of viruses like dengue fever and chikungunya by the mosquitoes between humans. So there's a very big program at the moment releasing Wolbachia-infected mosquitoes in order to try and interrupt disease transmission. So the first question we were interested in, which is the sort of rather simple one of how common is antiviral protection by Wolbachia? Is it just one or two strains, or 40% of insect species have Wolbachia? Does that mean that 40% of insects have some level of antiviral protection? So to look at this, we took a bunch of different Wolbachia strains which were found in different species of Drosophila, and we transferred them to this species Drosophila similans. So to do that, you suck the cytoplasm out of the embryo of one species and stick it into Drosophila similans. So we then had a panel of about of 20 different Wolbachia strains, all in genetically identical Drosophila, and we infected those flies with two different viruses, Drosophila C virus at the top and flockhouse virus here. And what we found was that 9 of the 20 strains provided some protection against viruses. So this is just a measure of protection. A value of 0 means no protection, and then the larger the bar, the higher the protection. So it seems that maybe half of Wolbachia strains protect against viruses. So therefore, if you make the sort of dubious extrapolation to all insect species, maybe 20% of insect species benefit from the antiviral properties of Wolbachia. And we've also repeated this using a similar panel of Wolbachia strains, but this time in the original hosts of the Wolbachia. And we find actually slightly higher proportion of Wolbachia strains protect the insects against virus infection. So this isn't just a sort of artifact of sticking them all into Drosophila similans. So there was some debate as to whether or not Wolbachia was providing resistance to infection, where it's preventing viral replication or at least reducing the viral load, or is it providing tolerance? So there was a suggestion that for some viruses, you got the same viral load, but the flies were just less likely to die. So we find that when we correlate our survival after viral infection against a viral titer, we find an extremely strong correlation for both viruses. So it seems that what Wolbachia is doing is it's preventing viral replication. And we now have some more sort of direct virological evidence for that. We also find this protection is very broad spectrum. Again, this is our 20 different Wolbachia strains, protection against flockhouse virus here and Drosophila C virus here. These are both positive sense RNA viruses, but otherwise are extremely different. They have really quite different replication cycles. And we find a really good correlation between the two things. And that sort of implies that there might be some sort of common mechanism underlying protection against these very different viruses. And we can do some statistics and control for the phylogeny of the bacteria, and that doesn't seem to be a confounding factor. So what we find is that, we then wondered why it was that the different Wolbachia strains varied in the level of protection they provided. So we measured firstly the density of the Wolbachia within the insects and correlated this to protection against the viruses. And regardless of which virus we use or how we measure, whether we measure viral titers or survival, we find an extremely strong correlation between Wolbachia densities and protection against viral infection. So it almost looks like any Wolbachia strain can protect the insect against viruses, provided you have enough of it, and you need an awful lot of it. If you take these sort of things up here and do some EM on them, the cells are absolutely crammed full of Wolbachia. There's a lot there. So you need lots of Wolbachia to protect you against viruses. And perhaps unsurprisingly, therefore, that in the absence of the virus, these highly protective Wolbachia strains tend to be bad for the flies. So here we've got hatch rate here against viral protection here. And what you can see is that the highly protective strains here tend to dramatically reduce the hatch rates of eggs. And we also look at other traits such as survival and fecundity, or pretty much any fitness component we thought would be pretty easy to measure. And we find that the most protective strains tend to be bad for the flies. So you get this very broad spectrum protection against viruses, but it comes at a cost. So that was, I promised you a talk about variation in populations. That was actually really variation between species, because those Wolbachia strains were originally isolated from different species. But with Luis Teixeira and his student Eva here, we've also looked at genetic variation within Drosophila melanogaster populations. And this is really primarily Luis's data, not our data. So the first thing we did with Casey Bergman in Manchester was we assembled a complete genome sequences of Wolbachia strains from within Drosophila melanogaster populations. And we just did this by scavenging sequence reads out of other people's genome sequences. So this is a phylogeny from complete genome sequences of about 188 strains. And you see that the strains all shared a common ancestor about 5,000 years ago. And there's two major clades of Wolbachia within Drosophila melanogaster. There's this big clade here, which I'll call the common clade, and this small clade here, which I'll call the rare clade. So what we did was we took 8 strains of Wolbachia from melanogaster populations and transferred them into an identical genetic background of Drosophila. And we took 4 of these from the common clade and 4 from the rare clade. And then we infected them with a virus, and this was, in this case, a flockhouse virus. So this is survival over 20 days. This is a control with no Wolbachia in light grey. It dies fairly fast. If you have the common clade, you benefit from a small level of protection. And if you have the rare clade, you benefit from much higher levels of protection. So you've got 2 Wolbachia strains circulating in the populations. The commoner one gives you a small level of viral protection. The rare one gives you lots of viral protection. So that's interesting, but it's particularly interesting in the light of an old study by someone called Marcus Riegler. And what he did was he went to old Drosophila stocks in labs, which had been collected right back from the 1920s up to the present day. And he looked at what Wolbachia strains they had. And he found that the old stocks tended to have the rare clade of Wolbachia, and the common stocks tended to have the common clade. So over the last century, there seems to be a replacement across the world of the rare clade was the common clade, and it's become the rare clade. So what's happened has been a replacement of the highly protective strain of Wolbachia by a strain which gives you much lower levels of protection. So that's slightly odd, really, because you'd think it's good to be resistant. So why is it that the low protective strain, the highly protective strain, has been replaced by a strain which only gives you low levels of protection? Well, it turns out that the difference in the levels of protection is again caused by a difference in Wolbachia densities. So the high levels of protection here are associated with high levels of Wolbachia densities. And it turns out that if you look at the survival of flies in the absence of Wolbachia, then at least in 3 of the 4 cases, you find that the highly protective strains are reducing the survival of the flies when they don't have a virus. So the high levels of protection, again, are coming at a substantial cost. And it's probable that maybe in the 20th century, the relative benefits of being resistant relative to this cost have changed for Drosophila, and that led to one strain of the Wolbachia replacing the other. So that's my 4th and final conclusion, that antiviral symbionts, they seem to be very common in nature. They're also a rather costly way to become resistant to viruses. We don't seem to see any obvious cost to our resistance genes, which we find otherwise, but we do find a cost to these things. And they can very rapidly change. So these are my conclusions overall. There seems to be a simple genetic basis of resistance with major effect variants explaining much of the genetic variation. This simple genetic basis was explained by directional selection, driving those major effect variants up to high frequency within populations. The genes tend to be rather specific in their effects. We see these allelic series with multiple alleles within a single gene, and they seem to be modifications to, at least in several cases, to existing antiviral genes. And finally, the symbionts are antiviral symbionts. They're very common, they're costly, and they rapidly change. So a variety of people have contributed to this work. So most of the people in black have actually contributed rather little, and the people to notice are the people in red. So Panita did the bit I showed on mosquitoes. The Drosophila work was primarily done by Mike McGuire and Chuan. The symbiont work was mostly done by Julien. And the final bit, a lot of the work was done by Luis Teixeira and Eva, who are based in Lisbon. And we're funded primarily by the Wellcome Trust and the European Research Council. So, thank you.